All right, so today we're diving deep into some pretty cutting-edge research. We're going to be looking at this paper, Bio-xLSTM, Generative Modeling, Representation, and In-Context Learning of Biological and Chemical Sequences.
Yeah, that's quite a mouthful. But I think the title kind of hints at the ambition here, right? We're talking about AI, but AI being used to understand the language of life itself, DNA, proteins, even small molecules.
And you know me, anything that says it can crack the code of biology, well, that definitely gets my attention. But I'm curious, like, where does this Bio-xLSTM fit into what's already out there?
Well, I think the paper starts by addressing, the big thing in the room, which is transformers. So transformers have been dominant in biological sequence modeling, but they have a weakness. Their Achilles heel is long sequences. So as sequences get longer, the cost of computation just explodes, and that's a major problem when you're dealing with like entire genomes or, complex protein interactions.
So even our AI superstars, they've got their
They
limits.
do.
So what makes this Bio-xLSTM different?
Well, think of it this way. Transformers are great at parallel processing, but they have a limited memory. Bio-xLSTM, on the other hand, is based on, it's called a recurrent neural network architecture. And what that means is that it has like a built-in memory, and that memory allows it to efficiently process much longer sequences, so it can capture those really important long-range interactions that we see in biology.
Okay, so it's got a good memory, but how does it actually work with all the different types of biological data? I mean, DNA, protein, small molecules are very different things.
Right, and that's where I think it gets really interesting. The researchers actually developed three specialized versions of xLSTM. There's DNA-xLSTM, Prot-xLSTM, and Chem-xLSTM.
Okay,
And each one is tailored for a specific type of data.
okay, let's start with DNA-xLSTM. What makes that so special for, deciphering the Book of Life?
Well, one of the key features of DNA-xLSTM is that it incorporates something called reverse complement equivariance. Basically, that means the model understands the inherent symmetry of DNA strands. So remember, DNA has two strands, and they're like mirror images of each other. So this feature ensures that the model doesn't treat a sequence and its reverse complement as two separate things. And that's really important for accurate modeling.
So it's almost like the model is learning to read the DNA in both directions at the same time, understanding that the information it's encoded in that mirrored relationship. It's pretty clever. But what about proteins? They're the workhorses of the cell. They're doing all kinds of things. So how does Prot-xLSTM tackle that complexity?
So Prot-xLSTM uses, it's called a homology-aware approach. And so what it does is it leverages the evolutionary history of proteins to understand their structure and their function. So by looking at the relationships between different proteins, it can identify patterns and then make predictions about how a protein will fold, how it'll behave, even if the structure hasn't been like experimentally determined yet.
So it's almost like the model is learning to read the evolutionary language of proteins, right? Seeing how they've changed over time to adapt and do all these different roles.
Yeah, exactly. And one of the things that's really exciting about Prot-xLSTM is that it's really good at in-context learning. So imagine feeding the model a few examples of proteins that have specific function, and then it can design new proteins with, similar properties. So you can imagine this revolutionizing fields like enzyme design or the development of new biopharmaceuticals.
Yeah, that's a game changer right there. I mean, that could really speed up the development of new drugs and therapies. Okay, so last but not least, we've got this Chem-xLSTM. What's the story with that one?
Well, Chem-xLSTM is all about small molecules. These are the building blocks of, everything from drugs to materials, and so it's particularly good at generating new molecules with desired properties.
So it's like a molecular architect that can actually design these molecules to order. So instead of just blindly testing thousands of compounds in the lab, you could use Chem-xLSTM to create like a short list of candidates that are worth exploring that could save, years of research and development time.
Absolutely, and like Prot-xLSTM, Chem-xLSTM is really good at in-context learning. So you provide it with some examples, and it can then generate molecules that have similar characteristics. So it's like teaching the model the rules of chemical design by just showing it a few examples, and then it can go off and create its own masterpieces.
This is all incredibly impressive.
Hmm.
But how do we know if these specialized XLSTMs actually live up to the hype? Like, did the researchers actually put them to the test?
Oh yeah, the paper is jam-packed with experiments, and the results are they're really compelling. So, for example, DNA-xLSTM outperformed some of the state-of-the-art models in several DNA sequence modeling tasks, like predicting the function of DNA segments or identifying gene regulatory elements.
So it's not just theory, it's actually delivering in real-world scenarios. What about Prot-xLSTM? How did that perform?
Prot-xLSTM also had some remarkable results. It really excelled at tasks that involve these long-range dependencies, which are so crucial for understanding how proteins fold and how they interact. It even surpassed some of the specialized protein models in some cases, which just shows that it's really good at capturing these complex relationships.
And what about Chem-xLSTM? Did it pass the chemistry test?
It aced it. It achieved top scores in generating realistic chemical structures, and actually it surpassed existing models in certain aspects. It even showed potential in a drug discovery task where it was generating molecules with specific properties that could be useful for developing new therapies.
So we've got these three specialized xLSTMs, each one tackling a different aspect of the biological world, and they're all knocking it out of the park.
That's pretty remarkable. But before we get carried away, what are some of the limitations that the researchers that they acknowledged?
Well, one limitation that they mentioned was the manual selection of what are called hyperparameters during training. And basically, because they had limited computational resources, they couldn't explore all the possible combinations of these settings that control how the model learns. So there's a chance that even better configurations exist out there, just waiting to be discovered.
So it's possible that with more computational power, we could squeeze even more performance out of these models. What other limitations did they highlight? Another interesting point was about DNA-xLSTM. They used a character-level tokenization approach, which means they treated each base pair in the DNA sequence as a separate token. And while that's a pretty common approach, it could actually limit the model's ability to capture higher-level patterns in the DNA sequence. So they acknowledged that this could be a bottleneck when trying to scale to even longer sequences.
It's like trying to understand a sentence by just looking at individual letters without taking into account the words that they form, right? You might miss the bigger picture and the other noteworthy limitations.
Yeah, they also mentioned just the inherent complexity of biological systems. So, for example, while Prot-xLSTM can get a lot of information from protein sequences, it doesn't explicitly take into account the 3D structure of proteins, which is obviously crucial for their function. So they suggested that incorporating, that structural information could further improve the model's accuracy and its predictive power.
It makes sense, right? It's like trying to understand a building by just looking at the blueprints without actually seeing the physical structure. You're missing a critical dimension.
Exactly. But, even with these limitations, they emphasized that this is really just the beginning, right? There's still so much to explore and so much room for improvement.
And that's what makes this research so exciting, isn't it? It's not just about the results that they've achieved, but it's about the potential that it unlocks. Like it's opening a door to a whole new world of possibilities in biology, in chemistry, in medicine.
I couldn't agree more. This research is really pushing the boundaries of what's possible.
Well, on that note, let's take a break, and then we'll come back and we'll dive deeper into some of the specific findings and discuss the broader implications of this research.
Sounds good. Welcome back. So, we've been talking about these Bio-xLSTM models, but I think it's worth diving a little deeper into how they were trained, because these models, they weren't trained on just any data. They were trained on these massive data sets that really capture, the complexity of biology.
Okay, so what kind of data are we talking about here?
Well, for DNA-xLSTM, they use the entire human reference genome.
Wow.
Yeah, that's over 3 billion base pairs. It's like giving the model the ultimate genetics textbook to study.
Talk about comprehensive learning material.
Yeah.
And what about for proteins? Did they go all in on that data as well?
They did. So for Prot-xLSTM, they use something called the Open Protein Set, which is a database with over 500 million protein sequences. So they wanted to make sure that the model had a really rich and diverse understanding of the protein world.
And what about Chem-xLSTM? Did they just kind of throw all known molecules at it?
Yeah, not quite. They used a curated data set that contains a diverse range of molecules, so things like natural products, approved drugs, and molecules from various bioassays. So it's more about quality and diversity than just, sheer quantity.
So it sounds like they were pretty careful to avoid that jack of all trades, master of none kind of situation.
Exactly.
Okay, but how do you even begin to train a model on such massive data sets? I mean, that seems like a pretty daunting task.
Yeah, it is. And so they use this really clever technique called context length scheduling during training. And basically what that means is instead of overwhelming the model with full sequences right away. They gradually increased the length of the sequences that it saw. So they started with smaller chunks and then gradually expanded it.
Okay, so it's like starting with like bite-sized lessons and then gradually working your way up to the full course load. That makes sense. So speaking of challenges, we talked about some of the limitations that the researchers highlighted in the paper. Can you expand on that a little bit?
Yeah, so one of the key limitations was that the hyperparameter selection process, that we talked about earlier, was still manual. And because of the computational constraints, they couldn't explore every possible combination of these model settings.
Right.
So it's possible that, there are even better configurations out there that just haven't been discovered yet.
So it's like, trying to find the perfect recipe, but you only have time to test out like a handful of ingredient combinations, so you might miss out on the absolute best one.
Exactly. And they pointed this out as, a potential area for future research. With more computational power, we might be able to find, even better ways to train these models.
Okay. And what about, the character level tokenization approach that they use for DNA-xLSTM? I mean, they acknowledge that could be a limiting factor, right?
Yeah. Yeah, so treating each base pair as a separate token, it might prevent the model from really understanding those higher level patterns that exist within the DNA sequence. It's like, if you're trying to understand a sentence, but you're only focusing on the individual letters without actually considering the words that they form.
Right, right. You might miss the forest for the trees. So it sounds like there's some potential for improvement there too.
Definitely. And, they also talked about, the challenge of representing protein structure. So
Yeah.
Prot-xLSTM is really powerful, but it only uses sequence data, right?
Right.
It doesn't explicitly consider the 3D structure of proteins, which is obviously really important for their function. So they suggested that, if you can incorporate that structural information, that could significantly enhance, what the model is capable of.
Yeah, it's like having this really detailed blueprint, but no way to actually visualize the final building. You're missing that crucial spatial dimension.
Precisely. And so, even though there are these limitations, the researchers were still very optimistic about, the potential of Bio-xLSTM.
Okay.
They see this research as kind of a launch pad for future advancements. There's still so much that we can do to refine these models, explore new applications, and really push the boundaries of what's possible.
Okay, so we've dug into the training process, we've highlighted some of the challenges, but now I want to talk about the big picture. Like, what are the potential real-world implications of this technology? So
Yeah.
let's start with personalized medicine. I mean, could Bio-xLSTM actually change how we diagnose and treat diseases?
I think absolutely. Imagine being able to analyze a person's entire genome and then predict, their risk for certain diseases or even predict how they might respond to different treatments. I mean, this could revolutionize preventative care and lead to much more targeted therapies.
So it's like having this personalized health roadmap based on your unique genetic makeup. That's incredible. And it's not just about predicting risks, right? With something like Prot-xLSTM, we could potentially design personalized therapies like custom designed proteins that target very specific disease pathways.
Yeah, that's really next level stuff. We're talking about creating therapies that are tailored to an individual's specific needs.
Yeah. What about drug discovery? Could this technology accelerate the development of new treatments?
I think that's one of the most exciting applications. Chem-xLSTM could be a total game changer for drug discovery. That ability to generate molecules with specific properties that could drastically reduce the time and the cost of finding new treatments.
So instead of, just blindly testing thousands of compounds in the lab, you could use Chem-xLSTM to kind of create that short list of promising candidates, and that would significantly speed up, the whole drug development process.
Exactly. And with those in-context learning capabilities, Chem-xLSTM could even be used to design molecules that target very specific proteins or pathways. So you could end up with, more precise and more effective therapies with fewer side effects.
It sounds like we're talking about, a huge step forward in the fight against diseases.
Yeah.
But, as exciting as all of this is, I'm sure there are also, challenges and concerns that we need to consider, right?
Oh, for sure. While we should definitely be excited about the possibilities, I think it's really important to proceed with caution. These are incredibly powerful tools, and we need to make sure that they're used responsibly.
Yeah, we need to think very carefully about, any potential unintended consequences, and really make sure that these technologies are used ethically and for the benefit of everyone.
Absolutely. And I think we need to have, some open and honest conversations about the potential risks and benefits and involve experts from all sorts of different fields. So, scientists, ethicists, policymakers. This isn't just a scientific endeavor, it's the societal one.
Okay, well, this has been an incredibly thought-provoking conversation. We've really explored a lot of the intricacies of Bio-xLSTM, from its training process to its potential to really revolutionize medicine and drug discovery. And I think it's safe to say that we've only just begun to scratch the surface.
Oh yeah, for sure. This is a field that's it's ripe with possibilities, and I'm really excited to see where this research leads in the coming years.
Now, for our final part, let's delve into some of those broader, like philosophical and ethical questions that this research raises.
I'm looking forward to that conversation.
So, welcome back to our deep dive into Bio-xLSTM. We've covered a lot, from the technical details to the potential applications, but I kind of want to step back now and consider the even bigger picture here.
Yeah, it's important to do that. I mean, this technology isn't just about incremental improvements. This has the potential to fundamentally change how we approach scientific discovery.
Absolutely. This research is sparking some pretty profound questions, about the future of biology, chemistry, and even our understanding of life itself.
Yeah, like for instance, in the realm of synthetic biology, imagine using Bio-xLSTM to design, completely new organisms with specific functions.
That's a mind-blowing concept, right? We're not just talking about understanding existing life. We're talking about creating new forms of life designed for very specific purposes.
Yeah, and that obviously raises all sorts of ethical and philosophical questions. What are the boundaries of creation? Like, what are the potential risks and benefits? These are questions we really need to grapple with as this technology continues to evolve.
And it's not just about creating new life, it's about understanding existing life in ways that we never thought possible. Imagine using DNA-xLSTM to really unravel the complexities of the human genome, identifying the genetic basis of diseases and developing very targeted therapies.
Yeah, the implications for personalized medicine are huge. We could actually move away from that one-size-fits-all approach to healthcare and move towards, truly individualized treatments that are based on a person's unique genetic makeup.
It's like having a custom designed medical plan just for you, tailored to your specific needs and your predispositions. But, it also raises these questions about data privacy and access. Who will have access to this information and how it will be used?
Those are critical considerations for sure. Like with any powerful technology, there's always that potential for misuse. So we really need to establish some very clear ethical guidelines and regulations to make sure that this technology is used responsibly and equitably.
And it's not just about the ethical implications, it's also about the philosophical ones, right? I mean, Bio-xLSTM is kind of blurring the lines between biology and computation. It's making us question what it means to be alive, what it means to evolve, what it means to create.
Yeah, it's really challenging our traditional understanding of life itself. We're essentially using the language of computation to understand and manipulate the language of life. It's a very powerful convergence of disciplines.
And it's happening so fast. This research, it's really just the tip of the iceberg. It's clear that Bio-xLSTM and similar technologies are just going to continue to advance rapidly in the coming years. And that's going to lead to even more profound discoveries and even more challenges.
It's definitely an exciting time to be in this field where we're witnessing a real paradigm shift in how we approach biology and chemistry, with the potential to transform our world in really profound ways.
It's a good reminder that scientific advancements aren't just about the technical breakthroughs. They're about pushing the boundaries of human knowledge and understanding.
And I think it's our responsibility to make sure that those advancements are used for the betterment of humanity and to address those ethical and philosophical questions along the way.
Well said. This deep dive into Bio-xLSTM has been a really incredible journey. We've explored the technical details, the potential applications, and the broader implications of this groundbreaking research. And I think it's safe to say that we've only just begun to scratch the surface.
Yeah, it's a field that's ripe with possibilities, and I'm really eager to see what the future holds. This is just the beginning of a new chapter in our understanding of of life and our ability to really harness its power.
Thank you for joining us on this deep dive. We hope you found it as fascinating and as thought-provoking as we did. Until next time, keep exploring, keep questioning, and keep pushing the boundaries of what's possible.